All right, we are ready to begin a new unit, and this is cells. So this is cell video number 1. I actually have another video loaded that is very long that goes into a lot more detail on each of the organelles. For this one, I'm going to try and keep it simple, knowing that you're going to go to your book, you're going to draw your own cell, and add more Cornell notes about what each one functions as. So I'm literally going to go clockwise right around these cells and just describe the function of each organelle. Here we have an animal cell, a plant cell, and a bacterial cell. So the difference right away, looking at them, we can tell there's a lot of organelles in common between an animal and a plant cell. There's of course going to be some in a plant cell that you're not going to find anyplace else, like a chloroplast. You usually recognize plant cells because they usually tend to be more square or rectangular in shape with flat sides, versus a lot of animal cells tend to be round or oblong. A bacterial cell lacks a lot of organelles. Usually, first of all, they do not have any membrane-bound organelles, so anything that has a membrane around it, they do not have. So let's start right here. Centrosome is an organizing center for microtubules, and it helps a cell divide. So centrioles, there are 2 of them that make up the centrosome. So 2 centrioles lie perpendicular to each other, and out from them will come spindle fibers. And if you remember mitosis, spindle fibers are what attach to the chromosomes and separate them to opposite sides. They are arranged in proteins they go around in a circle of 9. Cell membrane is what allows certain things to enter or exit the cell. We're going to have another video that completely just focuses on the cell membrane. A vacuole is a storage organelle. Ribosomes produce proteins. There's no membrane around a ribosome, and they're very, very abundant within a cell. Endoplasmic reticulum is, in Latin, it means like net within the cytoplasm. So it stretches out and it extends through quite a bit of the cytoplasm. Some of the ER is rough, meaning it has ribosomes on it, so it receives proteins that are made by the ribosomes, and it can transform them and package them into vesicles. A vesicle is just a membrane-bound organelle that usually goes from the ER to the Golgi bodies. Smooth ER does not have any ribosomes on it, hence the name rough. If it has the little bumps, the little ribosomes on it, it's rough. Smooth doesn't have any ribosomes. Smooth is responsible for producing different types of fats, phospholipids, oils, steroids, so something like cholesterol would be made in smooth ER. A mitochondria, you're going to see a lot of mitochondria in a cell that has to produce a lot of energy. So muscle cells have a lot of mitochondria. That's where cellular respiration happens. And more specifically, it's where aerobic cellular respiration occurs. So it's going to use sugar to actually get ATP. The nucleus, I'm sure you've heard that that's the control center of the cell. It's considered the control because the directions for building proteins exist in the nucleus. So the nucleus is where DNA is. The nucleolus is a region within the nucleus that is very dense, and that is usually where RNA is produced inside of the nucleus. Chromosomes are our segments of DNA that contain all of our genes that are found within the nucleus. And the nuclear envelope is the membrane that goes around the nucleus. It's 2 layers and it has pores. So that DNA is too large to leave through the pores, but RNA is small enough to leave through the nuclear pores. All right, Golgi complex, it sort of looks like a stack of pancakes. It extends usually from the ER towards the plasma membrane. So the Golgi bodies are what help receive vesicles from the ER, make some changes to those proteins or fats that are coming from the ER, and then package them up in another vesicle that will fuse with the membrane, and exocytosis will occur. Things will go out. Cytoplasmic plasm is the liquid portion of the cell, usually contains a lot of enzymes and things that are allowing certain reactions to occur there. All right, things that we haven't mentioned. Over in the plant cell, there is a much larger vacuole. The large vacuole helps hold up the structure and the shape of the cell. Chloroplasts, that's where photosynthesis occurs, and chloroplasts have a very unique structure with stacks of grana and a liquid called stroma within them. The stacks of grana are made up of thylakoid membranes, and that's where the pigments like chlorophyll are found in order to do photosynthesis. Ribosomes, again, is in both. Cell wall. Cell wall is only found in plant and fungus and some bacteria. So a cell wall for a plant is made up of cellulose. It is not alive. It is one more layer that substances have to go through to get in. It's not selective because it is not alive, but the fibers of it help hold up and give shape to plants. All right, down here in the bacteria, you see a lot of pili. Pili are these extensions. They're proteins that extend out. Ribosomes, of course, because you got to make proteins. Chromosomes, but no nucleus. The chromosomes are just within the cytoplasm. And capsule is another name for the whole interior of this cell. Sometimes the coating around it, we'll refer to it as a capsule, encoding the whole part with the membrane. Anything else that we left out? I feel like there's one more thing that's missing from here. It would be microfilaments. The cytoskeleton of a cell is that which supports it. So in a plant cell wall, I don't know why my board is going off. In a plant, the cell wall is giving the support, but there are also, in an animal cell which doesn't have that cell wall, there are small little tubes of protein, and those microfilaments or microtubules are what help give it additional structure and support. So I put up a couple other words here I just want to talk about. A eukaryotic versus a prokaryotic cell. Eukaryotic is like our cells. They're very advanced. They have membrane-bound organelles. So you will see nucleus, mitochondria, chloroplast, Golgi bodies, all of those things that have membranes around them within a eukaryotic cell. Prokaryotic cells are bacterial cells. Before there are membrane-bound organelles and before there are any nucleus, those are the types of cells. Very, very basic. They both will have ribosomes. All right, endosymbiosis. This means how did we go from a prokaryotic cell to a eukaryotic cell? So this idea of endosymbiosis, we've talked about symbiotic relationships. They believe that cells that were actually capable of doing photosynthesis were engulfed by another cell. So once the engulfing has happened, this cell has smaller cells within it. And those individual cells ended up taking on a role, living together, and now these are considered organelles instead of individual cells. So they all work together as one cell, and these organelles gave rise to eukaryotic cells. Cells that are capable of maybe doing photosynthesis within that cell or doing respiration within that cell. So symbiotic because they're working together. Something that they've been able to look at and prove is that mitochondria has its own DNA. If it has its own DNA, it's sort of proof that it could have existed once before as its own little individual cell. And then my last slide here, endomembrane system. If you look at a cell and you go from the nucleus on out, you can actually see components of the endomembrane. The membranes are made up of, here's ER, which is a very membranous system. It could have ribosomes on it, making it rough. The proteins made by the ribosomes actually get carried through this system until they end up in a vesicle. The vesicles fuse with the Golgi bodies, carrying those proteins along. Those proteins can be changed, maybe made into a glycoprotein along the way. Golgi body will modify it, sort it, package it again into a different vesicle, and that vesicle will fuse with the plasma membrane and exit the cell. So an endomembrane system means a system of organelles with membranes that help allow a substance to move out of a cell or into a cell. If something moves into a cell, the first thing that it fuses with, an organelle that we haven't mentioned yet, is called a lysosome. A lysosome has a lot of digestive enzymes inside it and helps break apart. So this is definitely going to be doing hydrolysis. It's going to break apart the components and allow the cell to then use maybe the amino acids that came from it or the monosaccharides or fatty acids, whatever was in what came into the cell can now be used by different regions of the cell. So I expect right now for you to go through the textbook, look up each of these different organelles and you choose what way you think will work best for you. Do you want to make a table where you list out each of the organelles, maybe what they look like and what they do? Or would you rather draw your own cell for yourself, label the organelles, and next to the label write what its function is? I do expect you to know the different organelles that we talked about today, be able to recognize them from a diagram, and tell me what they do.